Thank you, Swati, for a warm welcome. Can you just enable me to share the slide?
Yes, Dr. Austin, I think you are the host now, so you can take over.
Okay, thank you. Am I visible? Is my slide visible to all? Okay. Thank you. Uh, I'm audible? Uh, Mm. Mm. Is it visible now? Can you see the slides now? Right. So, as already Swati has introduced me, let me just start by introducing myself. I'm Dr. Afreen Inam. I'm an assistant professor at the Department of Chemistry, Jamia Millia Islamia. So, today I will be talking about medicinal chemistry and rational drug design for precision and personalized cancer therapy. So, first, any field which is related to drug design or medicine comes under the domain of medicinal chemistry. So, let us start with a brief introduction of medicinal chemistry first. Medicinal chemistry is not a standalone field, it is an interdisciplinary science which focuses on discovery, synthesis, and development of new drugs. And it brings together different domains like organic synthesis, biochemistry, chemical biology to create or modify molecules, to biochemistry, that is for disease mechanism and molecular targets, pharmacology, pharmacology to new drug interactions, toxicology to ensure safety, and computational studies to predict and optimize drug behavior even before the synthesis. This integration of diverse disciplines, this is what enables the medicinal chemistry to move from a biological idea to clinical useful therapeutic agents, making it central to rational drug design and precision medicines. With this overview, let us explore how all these components work together in drug discovery process. So first of all, let us talk about what is the concept of drug designing. Drug designing is a systematic yet creative process in which new drugs are designed on the detailed knowledge of the structure of the target, such as enzymes, receptors, and proteins. It is the core component of drug discovery and emphasizes on rational planning and optimization rather than trial and error methods. A key focus of drug design is the understanding of molecular interactions between the drug and its target, while we simultaneously optimize physiochemical properties such as solubility, stability, and bioavailability. Thus, the drug design represents a balance between scientific reasoning and innovation, which is aimed at developing effective therapeutic agents. Now, this slide highlights the scientific scope of medicinal chemistry in drug discovery. Okay, so here, despite significant advances in the rate of new drug discovery has slowed down, which is due to the stringent safety and efficacy requirements, complexity of rational designing, and the high cost of research and development. But still, medicinal chemistry supports drug design by, although it is being very challenging, still medicinal chemistry provides a chemical foundation for drug design by focusing on identification and synthesizing systematic optimization of biologically active molecules. These molecules may originate from natural drugs, screening libraries, or computationally designed scaffolds. Beyond synthesis, medicinal chemistry is deeply concerned with molecular mechanism of drug action, which includes how small molecules recognize and bind to the biological targets, the nature of their key interactions at the binding site, and how structural modifications translate these changes into biological activity that we describe as structure activity relationship. The ultimate objective of this discipline of a medicinal chemistry is what? It is the discovery of novel therapeutic agents, achieving which requires interdisciplinary skills. So, a medicinal chemist must integrate advanced organic synthesis along with biochemical understanding of the disease pathways and pharmacological evaluation of the drug responses. In this way, the medicinal chemistry act as a bridge between the converting the molecular structures into clinically relevant medicines. So before we get into the depth of medicinal chemistry and drug design, that is, let us just get aware with some of the terminologies which are used commonly in the medicinal chemistry. Most of you, all of you must be very much well aware of all these terms. I'm just giving a brief uh, view. So first, what we are talking about drug. What does that mean? It is a chemical substance that has already been proven to be safe and effective in humans for treating diseases. However, in early research, we don't call it as a drug, we call it as a hit. A hit is simply a molecule that shows some measurable activity in initial screening tests. Through these chemical modifications and optimization, the hit can be improved to a lead, which is further selected for optimization. If it shows better affinity, acceptable biological properties, it becomes a drug candidate suitable for clinical trials. Most drugs act by binding to specific biological targets, which are known as receptors, and depending upon how they interact with these receptors, they can be act as agonists or antagonists. That is either producing a biological response or they are blocking that response. Once we have a potential drug candidate, the next step is to understand how well and how safe they are. So potency, it tells us how much drug is needed to produce that effect, while efficacy tells us the maximum effect the drug can produce. These are often compared using the EC50 values. Safety index, safety of a particular compound is assessed using therapeutic index, which gives an idea of the margin between effective and toxic doses. From a medicinal chemistry perspective, one of the most important concept is structure activity relationship, that is SAR, which helps us to understand how the changes in the molecular structure affects its biological activity. And finally, we consider how a drug behaves in our body. That is how it is absorbed, distributed, metabolized, excreted. And now we also talk about toxic, what does the toxic effect it has on our body comes under pharmacokinetics. And in contrast to which, pharmacodynamics is the explanation of how drug behaves inside the body. What is the biological effect the drug is producing in the body? Now, I have shown in this slide, it basically summarizes the entire drug discovery journey from starting to an initial idea till the time it reaches and becomes an approved medicine. So it is a long, demanding process. Typically, 12 to 15 years, and it costs millions and millions to make one successful drug. Although once it is made, you can easily recover all that money also. So when we start with the target identification and screening, we followed by hit and lead generation, where medicinal chemistry plays an important role in optimizing its potency, selectivity, and safety. Only few optimized molecules, they move to clinical trials. Like in phase 1, they focus on safety. In phase 2 and phase 3, they focus on efficacy and validation before it reaches the final regulatory approval. And here I have shown a pyramid which shows that the biggest challenge is that from thousands to one which reaches the market. That is why rational drug design and precision medicines are so important. That is, they help in reducing these failures, and we can deliver the right drug to the right patient. Now, in this slide, it has summarized a typical in silico drug discovery workflow. How does we reach from thousands to a minimal point at laboratory level? We start with the pharmacophore model, which defines essential structural electronic features required for biological activity, such as hydrogen bonding, donors, acceptor, hydrophobic regions, aromatic rings, charge centers. These models are then used to screen large chemical database. And using this pharmacophore, we perform this database screening of virtual libraries, in which filters like Lipinski's rule of five, which are applied to ensure drug likeliness. And while this pains filters are used to eliminate assay interfering compounds, and this step dramatically reduces the number of candidates while retaining their chemical diversity. The shortlisted compounds then undergo molecular docking to predict their binding modes and their interactions with the target's active site. Then molecular dynamic simulations are used to assess the stability of these complexes over time. And in parallel, we also have ADME and toxicity predictions to eliminate candidates with unfavorable pharmacokinetic or safety profiles. And finally, the most promising molecules, they progress to experimental validation, where we synthesize the molecule, we confirm their structural confirmation is done by spectroscopies, then biochemical assays are performed in vitro, cell-based studies, and eventually in vivo evaluation, which are then further confirmed by computational predictions. What we had predicted before, they are all again validated after the experimentation. So overall, this workflow, this highlights the computational, how computational methods acts as a decision-making funnel, and they reduce cost and time by ensuring that only most promising drug-like candidates reach the experimental testing. So this is a brief about how we start in medicinal chemistry till we reach a experimental point. So now let us talk about where do we get the hits and leads from? The nature. The nature is our ultimate chemist. We all know that most of the drugs, as the data predicts, 50% of all the drugs and more than 60% of the anti-cancer drugs which are currently in use originally came from natural sources like plants or marine life. Nature gives us the lead, but we, the chemists, we turn it into a drug. Because these natural products might have some limitations, and exploring too much of nature is also destroying, it directly destroys the nature. Uh, so we as the chemist synthesize new roots in the lab, and we try to improve their properties, which allows us to manufacture them in large quantities artificially, and we get the medicine without having the harming or without depleting our natural resources. And here I have discussed two examples I've shown. One is of Taxol, which we know as an anti-cancer drug, which was used first in 1992. It was isolated from the bark of Pacific yew tree. Its first total synthesis was reported in 1994, which had multiple steps, and we are still working on to reduce the number of steps and produce more potent drugs for cancer, for which new therapies are coming every year. Another drug is discordamolide, which was isolated from Caribbean marine sponge, and it was also a potent anti-cancer agent. Now, as we have talked about the role of medicinal chemistry, and nature has given us the potent lead, like we have talked about Taxol. Here, we have a critical question that comes, that which part of these complexes or these structures which we get from nature are essential for activity, and which part of these structures can be modified. This question is addressed through the concept of structure activity relationship studies, that is the SAR of the molecule. This relationship relates the structure of the drug with its pharmacological activity. So here, the SAR of this molecule, it shows that this taxial core is essential for binding to microtubules. And any major change to this ring system leads to loss of activity. This C13 side chain is absolutely critical for anti-cancer activity. Removal of this or major modification in this chain can drastically reduce the potency. So, and we have specific functional groups also, hydroxyl and amide here. They play a key role in stabilizing microtubules. And at the same time, some positions are there like C7. These can be modified easily as they don't play a significant role in the activity. Okay, so by integrating the SAR with the rational design strategies, we move closer to the precision cancer therapy, where drugs are tailored to specific molecular targets. So, as we have talked about, we have seen that nature provides us the complex and potent lead molecules, and SAR helps us to identify their structural features which are important for activity. We now discuss the different approaches in drug design. First and historical traditional drug design was through analog based drug designing, that is designing of new drugs by structurally modifying the existing lead molecules or lead approved drugs. That is, we know a molecule, we know the structure of the molecule, we can just change the substitution, we can change the chain or a ring. A slight change in the structure can bring a desired give us the desired result, optimized result. Okay, in this strategy, what we do is we begin with a known biologically active compound. It can be a natural product, it can be an existing drug, uh, and we structurally modify that, which can be there can be two types of modifications: structural based modification, which we call structural analogs, and pharmacological based modification, which what we call pharmacological analogs. So I've taken example of morphine, in which, as you can see here, methyl group is bind to nitrogen, and it acts as opioid agonist. Whereas when this methyl group is changed with this alkene, here it acts as morphine antagonist. So structurally they are similar, except this functional group, they have a lot of structural similarity, but the pharmacological effect is different. And the pharmacological analogs are, as we can see, these three drugs, they are structurally absolutely different, but they have similar pharmacological property. Like in nifedipine, we have pyrimidine type of ring. Diltiazem, there we have seven-membered nitrogen sulfur containing ring, and here in verapamil, we have a tertiary amine linking two benzene rings. But although they don't have any structural similarity, but their pharmacological activity is similar. So now comes the idea to how do we optimize these structures, what changes, how these changes can be brought about. So to optimize these analogs in drug design, we can do that by bringing variations in the chain or in the substitution in aromatic ring. Like for example, I have taken adrenaline, in which again amine group is attached to methyl, and in isoprenaline, there is isopropyl group attached, and in salbutamol, we have tertiary butyl groups attached. So, what difference this brings? These changes in the structure. Adrenaline show potency for same for beta and alpha adrenoreceptors, while the other two they show better potency towards beta receptors. And we have talked about change in aromatic substitution. Like we can change the substitution from ortho to meta to para position. It reduces or increases the resonating or inductive effect, and here it shows how it improves the binding affinity through it increases the activity through hydrogen bonding. Just changing the position of OH from para to meta position, it has increased the activity. Another way we can optimize the analog is by extension or contraction in the structure. Extension means increasing the chain length. That is, if we have a molecule, say containing ethyl linker, we can extend the chain to propyl, butyl, and hexyl. We can lengthen the chain, or we can also contract the chain in the similar way. Or we can also do it by ring expansion or contraction. Like we have a benzene ring in a system, we can change it by uh, fused rings like indole, quinolines, or we can expand the ring from 6-membered to 7-membered ring, or contract the ring to 5-member. This also helps in better binding, having an extra group. So also I have shown some classical examples like cisplatin had severe kidney toxicity, and introducing in cisplatin, replacing these chloros with a dioxospiro ring. In carboplatin, it highly reduced the severe kidney toxicity. And another drug, that is nilotinib, it acts as super analog to treat leukemia to the patients which have developed resistance to imatinib. So this shows how structural changes helps us improve the properties. Another optimization in the analog can be brought through conformational rigidity. That is by giving the molecule, you're locking the flexibility of the molecule. Here, if we look at the conformational rigidity of this molecule, we have this is a single bond which is rotatable. Here we have induced rigidity by introducing a double bond. Rigidity can also be introduced using a ring system, or bulky steric constraints can be used. They make the molecule metabolically stable. And I've taken the example of combrestatin, which is a class of natural product. Here it has two forms, E and Z isomers. And as we can see, the Z isomer is active, E isomer is less active. Why? See, since this Z bond, we call it cis bond, it it binds to the consequent site of tubulin. It inhibits polymerization of the microtubules. It makes it rigid. This is a cis-Stilbene type of structure. This is the key to activity. Whereas in the E form, E isomer, the trans isomer, this does not fit or locks with the receptor. Hence, it is less reactive. Another approach in drug design is the rational drug design. That is, finding new medications based on the knowledge of biological targets. In rational drug design, this is a systematic approach for creating new drugs based on detailed knowledge of the structure of biological target rather than hit and trial. It uses the structure of and function of the target, such as enzyme, protein, and how does the molecule bind to it effectively. The key steps which include are target identification, that is using choose which biological molecule is relevant to which disease. Then lead discovery, that is finding the molecule that can best interact with that target, and then optimizing them. That is what chemical optimization we can bring and that structure to improve its potency, selectivity, or its pharmacokinetic features. And to carry out all that, we can use tools like crystallography, X-ray crystallography, computational modeling, and SAR studies. Two major components of rational drug designing are structure-based drug designing and ligand-based drug designing. So let us now discuss one by one what do you mean by structure-based drug designing and ligand-based drug designing. Is basically it depends on the three-dimensional structure of the biological target to which we want to design our effective drug. So for that, what do we know? We want to know the 3D structure of the protein target, which we can find out using extracrystallography, NMR, or cryo-electron microscopy. And then the potential drug which we are designing, or we already know, we have to optimize it to fit precisely into the active site. That is like a that is a key into a lock. And so for that, what this increases the binding specificity, and it also reduces the off-target effects. So here I have given a representation of how does structure-based drug designing work. That is starting from target identification and preparation. That is, target molecule is expressed, it is purified, and it is characterized using molecular biology techniques. Then structural determination of the target is done. That is 3D using X-ray crystallography, NMR, etc. After which we perform molecular modeling. like computational tools are used for which ligand design and optimization is done through virtual screening, through docking. Then, after multiple ligand keys are tested, which best fits into the lock, we come to the synthesis part, in which the lead compounds which are selected, they are synthesized in the laboratory. Their structures are validated through spectroscopic techniques, and further the drug target complexations are again analyzed by crystallography and NMR to confirm their binding mode. Next, they are evaluated for toxicology. Okay, and then the formulation, that is the optimized drug candidate, it is prepared into a suitable dose form, and finally it reaches the clinical trials. Another type of rational drug designing is ligand-based drug designing. It is just the opposite of the structure-based. You can say that, we can say that, because here the structure of the target is not known. Where the molecule is going to bind, we don't know that structure. And so in this approach, the information is only about the active molecule, the ligand which is available. So we analyze the active molecule, we identify the key features of that molecule which can be responsible for biological activity, and then we design the new molecule of similar activity. And the techniques which can be used are QSAR, that is quantitative structure activity relationship. It is a mathematical model which links chemical features to anti-cancer activity, or pharmacophore modeling, or virtual screening. So the advantages of ligand-based drug designing is that when we are not aware of the target structure, it can rapidly generate new leads based on the existing knowledge of existing active molecules. Now that following that, we have discussed SAR analysis and we have discussed analog and rational based designing. The next stage is how we move from hit to lead in drug discovery. So as I have shown here, multiple hits are generated from screening. They are refined into fewer and fewer through high quality lead compounds, which then enter into lead optimization, and which further move towards clinical to be clinical candidates. What is the goal of this hit to lead optimization is that it has multiple parameters, not just for improving the potency, but also keeping in mind various biological properties like selectivity, minimizing off-target effects, and also having improving ADMET properties. This stage is fundamentally, basically, SAR driven, structure driven, and we should know how structure is contributing to the activity. Then ideal lead is, therefore, it combines high target affinity, high moderate molecular weight, and lipophilicity. And whatever lead we reaches to, it should have a practical synthetic feasibility. The route to synthesize that molecule should be feasible. To explain this heat to lead discovery, I've taken one example. This is a real example. This experiment was performed in California Institute of Technology and Kansas University. This has shown the development of CB5083, which came to clinical trials. What they did was they had a large library of around 2 lakh molecules, which was screened through HTS ATPase assay, that is high throughput screening assay, leading to 925 primary hits which were identified. And these hits were again retested for the same assay, and they narrowed it down to 333 active molecules. And further, they expanded the orthogonal assays, manual validations, and to remove false positives, all that, and it yielded these 10 high quality hit compounds, and their IC50 values are shown. So these are chemical diverse hits which represented the starting point for the SAR analysis, where the medicinal chemists identify active scaffolds, understand their key functional groups, and decide which hits are suitable for hit to lead optimization. This shows the screening funnel, how large chemical space turns into a small number of validated hits. Now, out of those 10 hits, two were selected based on the micromolar ATPase inhibition and tractable heteroaromatic scaffold. These hits then served as a core structure for SAR exploration. Here, this shows how the key structural region targeted are which were modified were this aromatic core, these substituents tertiary amine R1 and R2, and this substitution R3 region, allowing them to tune the potency, binding interactions, whatever can be improved through. So they reached to a compound ML240, 240, and this is the synthetic scheme, which shows how an efficient rule of rapidly synthesizing these molecules, in which the dichloropyrimidine ring is there. This reaction is carried out, and overall, this demonstrate that rational SAR and synthetic accessibility transforms hits into a viable lead. And now, by using this ML240, they optimized it and they made it into CB5083, which has improved properties and had gone into clinical trials. What all they had using this ML240, they had the chemical starting point, and by structure guided lead optimization, they improve the binding interaction with the ATPase, reduce its lipophilicity, they enhance the oral bioavailability, and we can see that IC50 value of this was 815 nanometer, which reduced to 11 nanometer, maintaining the molecular weight and C-log P values. It was the first in class orally available P97 inhibitor. It advanced to human clinical trials. So, as now we have talked about medicinal chemistry, drug design. So, what is the precision and personalized drug? As we know that interpatient variability often causes the same drug to have different efficacy or toxicity profiles. This precision and personalized medicine, this aims to overcome this by integrating patient specific molecular information into drug development. So here, this first panel, it shows drug discovery and multi-omics profiling. This is the process which starts with drug discovery, target discovery using multi-omics profiling to analyze genomics and proteomics data assisted by AI algorithms to identify driver mutation, gene fusion. These molecular signatures, they allow to predict drug and target interactions, its druggability and potential resistance mechanism. We define this as the molecular vulnerability of the disease at individual level. Then this is the part which the medicinal chemists play, that is the rational drug designing and functional validation. Once the target is defined, rational drug design comes into play. The medicinal chemistry and computational modeling, this helps us design high affinity ligand that interacts with the target, same as lock and key optimization. Then in the end comes the precision application and personalized therapy, that is tailored to patient. That is, we integrate companion diagnostics, biomarker profiling, and pharmacogenomic data to select most effective therapy. That is the ultimate goal of this precision therapy, is right drug to the right patient at the right time. This workflow, this demonstrates how medicinal chemistry interfaces with the genomics, pharmacology, and computational biology to drive rational and patient-specific drug discovery. So, as now we have talked a lot about medicinal chemistry, drug design, structure-activity relationships. So, we have targets for cancer therapies that can be enzyme-based target therapy, and it can be immunotherapies, it can be combined therapies. So, what are target therapies? Is the type of cancer treatment that targets proteins that control how cancer cells grow, divide, and spread. It is the foundation for precision medicine. As a researcher, we learn more about DNA change and protein that drives the cancer. They are better able to design the treatment for that target protein. So I will be discussing a few of these. Like I have talked about tyrosine kinase inhibitors. Mostly, I will be explaining how the structure part plays the role. We have taken example of osimertinib and ivazanib. Osimertinib is a third generation drug, oral EGFR TKI specific. It binds irreversibly to mutant receptor via covalent interactions, offering enhanced potency and reduce off-target selectivity. If we talk about its structure, activity relationship, it has acrylamide warhead. This acrylamide warhead, it plays a crucial role in covalently binding to cysteine residue of EGFR. Then we have this methoxy group. It is positioned on the aromatic ring. It selectively blocks the wild type EGFR. So it improves the interactions and it improves the selectivity. Then we have this meta-substituted amine aniline ring. It is meta-substituted aniline ring. So it reduces the metabolic degradation, enhancing the stability of the drug. And this pyrrolopyridine scaffold, since this is a third generation drug, earlier drugs have indole. So it had shown better, superior activity as when we compared it to indole. Similarly, we have tivozanib here also. It has quinoline core and it has urea type linkages, which enables dual hydrogen bonding with VEGFR2, providing strong kinase binding. And incorporation of this chlorophenyl ring, this facilitates hydrophobic interactions, improving the binding affinity. Another targeted therapy is the kinase inhibitors. Uh, Main structural features include hydrogen bond donors, acceptors for hinge binding regions. Here also, if I talk about their structures, I have taken two drugs, palbociclib and abemaciclib. Palbociclib, it is characterized by the presence of this piperazine ring, which significantly improves the aqueous solubility and it helps in salt formation for formulation purposes. It has a tertiary amine in this piperazine ring. This enhances oral bioavailability. And this pyridine ring, which is attached to this piperazine, this is crucial for binding CDK selectivity. And this cyclopentane ring, it contributes to hydrophobic site filling, enhancing CDK6 inhibition. So now about another therapy which I have discussed is combination chemotherapy, which is the use of multiple agents with different mechanisms. The combination of sotorasib with vasitibinix is used. That is, it is the combination which was approved in early 2025 for the treatment of adult patients with metastatic colorectal cancer, but who have already prior received chemotherapy. It is this sotorasib. This is a covalent inhibitor targeting the rat sarcoma gene mutation, where the amino acid glycine is replaced by cysteine mutation. While this the drug with it is combined is vasitibix. It is an anti-EGFR monoclonal antibody. This dual targeted approach, this addresses the activation of EGFR signaling and it often limits the efficacy of class inhibitors. If I talk about the structure of these two drugs, here the sulfonamide, this group sulfonamide, it enhances the binding by hydrogen bonding, and the lipophilic tail contributes to this is the lipophilic tail. This contributes to improved cell permeability and oral absorption. In encorafenib, the pyrazolopyrimidine, this rings this scaffold with sulfonamide. This helps in targeting the ATP binding. Then let us talk about some approved FDA-approved drugs into clinical trials. They are targeted small molecules. They are designed for specific oncogenic target. Here I have discussed, given the structure of two, divarasib and glacirasib. Divarasib, they are KRAS inhibitors. And if we talk about their structure, activity, relationship of this divarasib, it has highlights the importance of this 2 amino, 4 methyl, and this 5 trifluoromethyl, this ring, pyridine ring, this moiety, and at C7, this enhances the binding of cross switch pocket. This properly orients with the acrylamide, this warhead. It acts as a warhead for covalently targeting the cysteine. This also maintains the binding pose across the GTPase supporting scaffold and flexibility. These findings emphasize the need to optimize both reversible binding and warhead reactivity for selective KRAS inhibition. So there were changes. It had certain drawbacks, so the changes were brought about in the new drug, that is glacirasib. In this, this is another irreversible KRAS inhibitor. This is in clinical development. Um, it has selectively binding to the and stabilizing GTP bond and to the inactive form of KRAS. It has this 1,8 naphthalidine 3 carbonitrile, 3 carbonitrile. This is 3 carbonitrile scaffold, which anchors to the switch pocket 2. While it is precisely, it is in position with this warhead again, which binds to cysteine 12. Further, this SAR refinements improve the solubility, reduce the metabolic liabilities, enhance their pharmacokinetic properties, resulting in a potent, well-balanced clinical candidate for KRAS mutant cancer. This glacirasib, this represents an optimized inhibitor with nano molar potency, and it increases. This increase its selectivity to 500 folds, contrasting with the earlier leads. Then let us talk about a new approach that is trending latest, the PROTACs, that is proteolysis targeting chimera technology. They induce the target protein degradation instead of inhibition. So I have taken the example of rich galutamide. This is a novel AR degrader which uses a PROTAC technology. This PROTAC-based AR antagonist and a degrader, this was developed to overcome the resistance in metastatic castrate-resistant prostate cancer. Here, the PROTAC, this is formed by combining three optimized components. The three components are the high affinity AR ligand, that is, and then the E3 ligase recruiter. E3 ligase recruiter, this give the efficient degradation, influencing the kinetic and target effects, kinetic and off target effects. And then this optimized linker, this linker, its length and its polarity, this influences the ternary complex formation and the cellular activity. So this is all about the type of therapies, cancer targets, which are currently in use. This shows the structures of the FDA-approved drugs. I've taken examples of tyrosine kinase inhibitors here. As you can see through the structures previously discussed and this slide, that there are heterocyclic rings which play the key role. Especially we have seen nitrogen-containing heterocycles, pyridine, piperazine, morpholine. So it is observed that heterocyclic rings a crucial role because they also help in hydrogen bonding. Easily, they have a lone pair of electrons. Keeping all these points in mind, we medicinal chemists design our molecules. So now here I'm going to present one of my molecule which was designed in my laboratory, that is hydroxamic acid base carbonic anhydrase inhibitor. This was designed by combining the features of known clinical candidate, this SLC 111 and some maplin C. So from here we have chosen this part, the zinc binding group, which were, and here the oxime hydroxamic acid group was introduced. This group act as a claw that grabs the zinc ion in the enzyme's active site, which helps in effectively stopping its activity. We have this linker and this tail. This helps the drug to fit into the enzyme and stabilizing the binding and improving its potency. So the synthetic route, keeping all these points and expecting it to show good results, the synthetic route for this was designed, and these compounds were synthesized in my laboratory. There were four key steps. Uh, First, we had taken isonicotinaldehyde, that is pyridine carboxylaldehyde, which reacts with hydroxylamine in presence of ethanol for 10 to 15 hours to produce this enzyme, which was further reacted with N-chlorosuccinamide NDMF to the chlorosubstituted oxime, which further reacted with these piperazine sulfonamides to give the target molecule. Here we had sulfonamides, here we have oxime and this pyridine linkage and this linker. 10 such derivatives were synthesized in our laboratory. Not alone this, similar type of compounds containing indole and sulfonamide linkage, which are linked via chalcones, such compounds were also synthesized. Another series was synthesized containing hydrozoles, in which indole and sulfonamide were linked through NNH bond. And morpholine was used instead of piperazine here. And these were the one which I have shown the synthesis of. So these compounds had shown they were potent towards cancers associated carbonic anhydrase enzyme. They showed good toxicity, cytotoxicity profiles, binding affinities. The results were quite good. The cancer studies were performed on HEK cell line and MCF7 cell line, breast cancer cell line. And we have published 3 papers in ACS journals from these 3 series. So, to conclude, I can say that to sum up all these studies, can say that the new study that highlights how the structure-based medication design can drastically speed up the process of finding and developing new drugs. And using these comprehensive structural knowledge and biological target knowledge, structure-based drug design, it provides a logic and an effective method for creating new medicine with improved potency, selectivity, and safety profiles. So I would like to conclude as as medicinal chemistry provides a molecular framework for precision and personalized medicine by enabling rational and target-specific drug design. Advances in structure-based and ligand-based drug design allows the development of therapies tailored to specific molecular targets, pathways, and disease subtypes. And precision method shifts from one drug fits to all to right drug to right target to right patient, improving the efficacy while minimizing the toxicity. And overall, the medicinal chemistry plays a central role in translating the biological insights into safe, effective, and personalized therapeutic agents. Last but not the least, I would say the precision medicines begin at the molecular level, but rational drug design is its driving force. Thank you all for giving your time and listening to me. I think I finished quite early.
No, it's absolutely fine. Thank you so much. We can take some questions here, right, Swati? We have got some time.
I think either they are already well aware with the topic, as they have been involved with this from past 4 days, or everything was beyond the
That
scope.
was very
So I,
clear.
I had actually kept it to the basics only.
Yeah, but I have seen that they warm up a little bit later, so don't worry. You might see the questions later on, and you can answer them as we go. So we will start at 12 with my lecture. I'll put up my current slide, and please feel free to ask any question that is related to the subject, or this is also an opportunity for asking a career level questions, because we have got some undergrads here as well as graduate students, so you can ask. And this was a very befitting lecture, Dr. Afreen, because we had a recently concluded Gyan course, which was on drug discovery and delivery. So your lecture connected the two Gyan courses that we conducted here. Um, Thankful to you for that. I think there's a question here. Swati, can you take it up?
See, what we can say in personalized cancer therapy. See, as I have explained already, drug design has played an important role, and a molecular this heterogeneity. When we get into the depth and we relate the structures to whatever type of cancer it is, we come to know the optimization the structure can bring. We can optimize the structure to personalize it to particular therapy depending upon the different targets and the already present drugs. See, I am not into much into the biology of all these. I am a chemist, so I think I can just say that combining the knowledge of both the drug and the target can help you in this to get a better personalized drug for cancer.
I can take up that question, Nilofer, because the molecular heterogeneity affects the finding of the target. That makes the life of the hunters for these cancer targets difficult. As I told you in my lectures, these are moving targets, plus they are different from one person to another person. But once the target is identified, then the drug design is not designing a drug from scratch for that particular person. Rather, we try to see what has already been designed and what has already been available as FDA approved drugs or libraries that we can bring into for helping this particular patient. Tumor heterogeneity or molecular heterogeneity is a problem for finding cancer drug targets, but once target is found, there's no problem with the drug. Unless there is something very, very rare, that is, that there is no drug or molecule available to target that. Does that answer your question, Nilofer? Right. So I think we'll just take it.
I think it's already 12 for 3 4 minutes. You can take a tea break or something if you want. Thank you, all of you. I'll just take your leave now.
What's left? We have been sticking to the schedule.
Thank you so much. Thank you, all of you. Thank you for, I would like to thank Professor Aziz for giving me this opportunity to speak today in front of you, all of you. Thank you, sir. And thank you for answering that question also on my behalf. Thank you.
By the way, there's another question by Fatima. Can you take that?
Yeah. How do you select the lead compound and what factors are? Okay. See, I have already discussed. We decide the lead compound by screening through computational studies. That was there in my slide in the very beginning. I said we from the library of compounds or from the already drugs which are the nature has given us or we have already we have so many drugs in the market, medicines in the market, they act as a lead for the future designing and optimization of the compound. Like I have taken one example of Taxol, that it was extracted from plant, and later on its total synthesis was designed, and now also whatever drugs for a particular disease or an infection is present in the market, we can take it as a lead compound. Or I would give you an example like for malaria, uh, the plant quinine was used, from the bark of that quinine was extracted from some plant. So later on, when we develop the drug for malaria, whether you talk of chloroquinoline, amodiaquine, all those have structural features similar to quinine, which was extracted from a plant. So nature, first of all, nature acts as a base to start from. That is from where we start. And the drugs which are already in market and the drugs which have already been synthesized, which are FDA approved, they set the base to new lead optimization. I hope I have answered. Thank you. Anyone, any more questions, please? Thank you.
All right. My slides visible. Thank you so much, Swati. Once again, very good morning and Assalamu alaikum to everybody. And I see the attendance is quite good. Aligarh is pretty foggy and it's a bit cold as well. I don't know how is it in Delhi. Starting enough. So today, this is not going to be an education lecture, rather it's a concluding one where I would like to summarize some of the stuff that we have learned so far, and also some recommendations to the policymakers and to the universities and to the students and participants here about how to proceed from here. The take-home messages in the 5-day course, what we talked about, what we discussed, what we worked upon, that how do we take it forward. So I would try to do that in a few spots. My last lecture. So if you have any questions or any concerns, I'm available now or later on as well. So just a recap that we started with a new, not new, but I would say a rare concept about using multiomics for finding the therapeutic targets. As I had said in the beginning, that we always knew that, yes, this is something that is relevant, but we didn't know how to do it. So when the genomics and transcriptomics and the proteomic technologies started to come up, we started doing it on the high throughput scale. We started to be successful with all the 3 billion bases that were known as the components of DNA. Once it was possible, then yes, we thought, why not we start integrating them? So that's how multi-omics is picking up. And still, I told you that there is a limitation on the data analysis part, that we still struggling to make sense out of all this data that is being generated in terabytes. So I hope you agree with me that cancer problem is like a jigsaw puzzle that we need to put together for finding the right therapeutic target. If we are not doing it, we are missing the picture, and we are not going to improve significantly in terms of the cancer care, in terms of making the life of these cancer patients better. And obviously, we have been more successful in preventing cancer. The early diagnostics, the liquid biopsy-based diagnostics, then the prevention strategies, the work on the lifestyle factors, reducing obesity and reducing other compounding factors. They have all been able to bring down the incidence, but still, once we diagnose and once it's progressive, the cancer has still have a dismal problem in terms of treating it. So we must uh, embrace or acknowledge this idea that we should look around the cancer research problem in its entirety and not on a just a piece. This is the snapshot. This is a summary figure that I showed you earlier as well. That what we are doing here, we are trying to generate the data from patient samples. We are trying to generate data from cell lines. We are trying to generate the multi-omics data from even the liquid biopsies. And once we have got this data, the genome, epigenome, transcriptome, proteome, metabolome, and there are other omics as well, we put the latest in the data analysis tools, and then we are able to get either biomarker discovery or we can find specific targets. And we can also look into the mechanisms of how these cancer cells are changing and how these cancer cells are shifting their target. The problem of drug resistance is there as well. Once a person is cured, again, the cancer comes up, and that is even more lethal because the drugs are no longer working. So that's why we need it for all these applications, and it has to be in a personalized manner and personalized treatment plan. This is how this is like a roadmap that we are trying to follow, and we are trying to develop evidence for every step of this paradigm. Excuse me. So a big help has come from the advancements in the machine learning and artificial intelligence technologies. All of you know that how disruptive it has been. It has changed the way we are doing our work. There's nobody who's not using ChatGPT or Copilot or Gemini. In fact, many of us have used it for preparing for this course and presentations. So this is something that is integrating into our lives, and it is also helping to integrate the multi-omics. And because there's no other way. No human mind can put together billions of data points and find a pattern out of it. It's only the machines that can do it. Yes, we tell the machines how to do it, but we can't do it. That's as simple as that. But the real challenge is to find out a use of these information. That how do we use it for patient stratification or patient characterization, or using it for developing biomarkers, or even for the diagnostic purposes in digital pathology. And the quest for finding out the mechanism continues, because what all we are talking about is based on some fundamental basic research that has been done over the decades. We shouldn't forget that this is the backbone of all the science that we are trying to put into the clinic. We are trying to help the patients, but it is the work of decades of basic research where we were able to know the mechanisms of these genes and the cellular pathways, the signaling pathways, or if you talk about the epigenetics, or you talk about the metabolism, or you talk about the drug. There's a lot of basic work that has gone into making the platform where these things are now feasible. You heard from Dr. Asim about different aspects. I think he has a lot of basic stuff covered here, and you learned about how the cancer is induced, what are the pathways involved, and how we can target those pathways. So these are some of the snapshots from his slides, that cancer could be a result of multiple mutations, which are accumulating over the time, and then it is reaching to a threshold, which takes the shape of a full-blown cancer. And while these mutations, which are actually errors in the DNA, while these errors are happening, there is error correcting machinery found inside our cells. This is the DNA repair machinery, which is also active. So it's a race between the DNA repair machinery and the insults or the errors that are happening in the DNA. Whichever wins causes the effect. If the mutations they win and DNA mismatch repair or any other repair loses, then we see the accumulation of these mutations. We have, like I talked about the example of the whale. The whale has got a very proficient and very efficient, fast DNA repair system. That's why even such a big animal, and there's a lot of dividing cells there, they don't get cancer. You want to ask Can you see my screen? I think there was some. Hello. Can you see my screen now? Okay. And is the slides are visible? Let's do that. How about now? Wait a second. All right. Is it okay now?
Yes, yes.
Okay. Can somebody please help me with telling him where did I leave? Where was the interruption? Swati, did you see this slide? Hmm. Anyways, so let me get ahead with this. Oh my God, that was long back here. Okay, so this is something that I was doing a recap for Dr. Asim, that we learned a lot of things about androgen receptor, prostate cancer, and how does RNA DNA repair work, and these mutation progression and mutation accumulation leading to the formation of the cancer. And we also learned about role of hypoxia and thrombosis from Professor Zahid Ashraf. We learned about metabolic aspects from Professor Hamid Ashraf, epigenetics from Dr. Siddiq, and recently, today you heard about preparing the drug libraries from Dr. Afi Anam, which is a very good fit between in the form of concluding this course that we are doing in finding the targets But once the targets are identified, we definitely need these drug libraries for going to the next level of clinical translation. So we have learned about all these things, and I picked up from this one of her slides, and the last here, the last message here about the precision medicine begins at the molecular level and rational drug design is the striving force. It's a wonderful conclusion because we, even if we find the target by using multi-omics and machine learning and alternative and artificial intelligence, what are you going to do with that unless we have the right molecule, right drug to target it? That's only when it becomes clinical reality. So we need definitely the help of these rational drug design and chemists to make it a clinical reality. Now, how a message for all the participants here that how are we going to do it? It's about collaborating effectively. That we should be able to come out of our boxes and shake hands to collaborate. Leave our insecurities, and for the students, don't think that I am in a particular course, so I cannot talk about other things. No, you should be opening up to collaborate effectively for the researchers, for the scientists, for the doctors. We should also be leaving our comfort zone and at least reaching out to the people from other disciplines who can help us out. There's no point in reinventing the wheel, rather we should be shaking hands to make things more accelerated. Thinking out of the box, and I think here reading some of the science fiction novels would help, because while we are working on a specific problem, it's very difficult to come out of that focus and try something new, but this is definitely needed. And when I started talking about this multi-omics about 10 years ago, people were thinking that I came from Mars. They thought that I have been talking about something very out of the world stuff. But now you can see that the publications are coming in, people are embracing this idea, and it's now even a clinical reality in some countries, in some areas of those countries. Some hospitals are adopting it for doing multi-omics. Longitudinal studies are coming up, where individual patient reports are coming in, and they are being recommended based on the multi-omics paradigm. So thinking out of the box would definitely help you in looking something extraordinary and looking into the future. Mentoring is one of the very important aspects. Get mentored, because and mentoring is not something that is to be done only for the students. Even when I relocated to India, I looked for mentors who can tell me about the Indian science system, how the funding is happening, how things work here, how collaborations work here. So there's no age or stage for getting mentored. Rather, whenever you feel like you need something that that needs mentoring, find a mentor and get mentored. That's always helpful. Now, since I have to talk about the framework as well, so I would suggest that in our university, we need to have more efforts so that we can contribute to the national and international efforts in finding cancer therapy targets and curing cancer. And so we need more efforts in teaching as well as research. And we need more people even to work in cancer biology to contribute to the international arena. We also need more industry academia connect, which is totally missing here. That even if we have got a target, even if we have the drug molecules available, who is going to test them and who is going to commercialize them, who is going to put them for the patients. So we definitely need more industry academia connect. And we also need it more for the funding of the research, because now the government funding is really, really difficult. Yesterday only, the Anusandhan National Research Fund, they sent their results for research grant proposals, and there was a bloodbath. There was hardly very few projects that got funded. So we need more industry academia connect to do that. More consortium efforts, and this is what we have started at Aligarh Muslim University. We have made a consortium with the help of 7 different departments. So there's a problem. The problem here is cancer for us. And we are trying to develop a collaborative exercise with the help of different expertise. I think such consortiums are needed in other institutions as well. And if there's any help that we can extend to establish those consortium and help them with the functioning of these consortium, we'll be happy to do that. And we have to push for this concept of precision and personalized medicine, because so far I think we are living in that archaic stuff where I see the funding priorities are based on the type of cancer, head and neck and lung and breast, but we are moving from that concept. We are moving towards precision and personalized medicine. We are moving towards not the tissue origin, rather the molecular origin. So we need the funding authorities to acknowledge this concept of precision and personalized medicine, so that we can have better outcomes, better societal benefits, better benefits in terms of the health of the people. So we have to think about it, because there's a misconception that it's going to be very expensive. Rather, when you talk about the precision in personalized medicine, it could be more economical, because we are not allowing unnecessary tests to be done on a patient. We are not allowing any unnecessary drugs to be given to the patient. We are not allowing any unnecessary surgeries to be done on the patient. If you look into the statistics, a lot of unnecessary work is done on the patients just because it's hit and trial. They don't know. They don't know the target. They don't know the person, and they are wasting their own resources as well as the resources of patients, causing them problems, mental trauma, and other things. So don't think that multi-omics is something very fancy and it cannot be done. It's very expensive. No, it is pretty much doable, and it should be done. Unless we do it, we are going to be going in circles, and there might be no solution to it. And I'm sure this technological improvements will bring down the cost. I have shown earlier slides that there was $1 million genome sequencing done in 2001. Now that same genome can be sequenced for less than $1,000. So when the technology improves, you can see that in case of cell phones, the electronic gadgets that you're using now. Once they were so expensive, not within the reach of a common person, now everybody has it, everybody can afford it. I believe that once technological improvements will be there, it will bring down the overall cost of precision and personalized. And definitely, since this course is focused on finding the therapeutic targets, we have to think differently. We have to bring a paradigm shift in how we identify targets using multi-omics approach. This is another slide that I had shown you earlier, that we can also use the same approach for developing biomarkers by using the existing data sets and then putting them into the integrated analysis pipeline, validating them using the in vitro or in vivo or ex vivo stuff, and we can get multiple biomarker discovery. Biomarkers are essential for practicing precision and personalized medicine. There is no, it will be very difficult to do precision and personalized medicine without biomarkers. Biomarkers are definitely essential for doing these. So this needs to be developed simultaneously while we are developing the therapeutic target. And keep in mind, the final outcome has to be precision and personalized medicine that saves lives. We are not doing just for the sake of publishing papers or doing academic exercises. Rather, we have to keep in mind that we have to save lives. And this is what will give us the passion and to work day and night towards this outcome. With that, I think I've come to my last slide about the acknowledgement. And this acknowledgement is, I'll start with our Vice Chancellor, Honorable Vice Chancellor, Professor Naima Khatoon, and the chief guest that we had. I'm summing up the two GAN courses here, so you'll see the names from previous one as well. We had Syed Ahmed, who was the CEO of Tech Invention, and Professor Zahid University, Professor Mohammad Asim, the principal of Zakir Hussain College of Engineering and Technology, Professor Sar Ahmed, the director of the Nanotechnology Center, and Professor Jahangir Watsi, our national coordinator for the GYAN course in AMU. And I would love to thank my co-faculties, Dr. Mohammad Zainuddin from Jubilant Therapeutics, Professor Hamid Ashraf, Professor Dev, and Dr. Afi Anam. And a big thank you to my PhD students, Swati Acharya and Sayyida Shari Zafar. They have done a lot of work. In fact, they were the backbone for and encouragement for me to do these courses. Without them, it would not have been possible at all. And organizers, Dr. Kasid Anwar, Dr. Sayyid Abdul Ahmed, and Dr. Salih Naseem, they have worked tirelessly to make sure that everything goes well. The previous one was in person. This one is online, and both of them had their own challenges, and both of them had got their own problems. So I would give a big thank you to the organizers, and definitely the peripheral setup by the grant section finance officer. This is all definitely acknowledged, and this is needed to do these courses, finance officer, PRO, polytechnic staff, learning garden, and everybody who supported. And last but not the least, uh, the main focus and component of this whole program is about the participants and volunteers. So thank you for attending this course. Thank you for registering this course, and I hope you go away with some messages, some passion, some fire in you that can help you in saving lives that are being destroyed due to cancer. With that, I would end my presentation, and I would love to take any question or discussion or so I'll leave this. There's something from Padmavati. I have a question regarding successful grant applications. Do you think including meta-analysis data as a preliminary result would increase the success rate of the proposal? I would say this is looking to be the future because a lot of data is available and we don't need much resources to analyze those data. So please go ahead and publish those results. Meta-analysis are more powerful than the original research articles because they are built up on multiple publications. So the evidence that comes out of meta-analysis is stronger. But I don't know how the funding authorities take it. Probably they might not consider it as a valuable exercise. But if I'm in the funding authority, I would definitely consider it as a valuable contribution. But the wet lab stuff is more and more difficult because of the funding crunch as of now. So we should be shifting towards data analysis, and that's very, very valuable everywhere. Okay, so let the audience warm up to and ask questions. So we keep the session live until 1 o'clock, and I'll be hanging around here. And then we also have the exam from 3 to 4. And this exam, Swati, can you tell us about the conduct of the exam once again, please? So at 3 o'clock, only the participants will sign in with their registered email ID. Yes, and there will be 100 questions out of 115 that needs to be answered. Yes, and all these questions are from either my slides or Professor Dr. Asim slide. So the questions have been prepared from only these two sources. They're pretty simple and it's a good exercise. And if you want to take the credit for the course, the exam is mandatory. If you don't want it, then yes, you can stay. Okay, so I think Padmavati, you are talking about the I want to write a project proposal based on the omic studies. What are the factors should I consider to get a successful funding? So the best factor is to tie up with somebody. If you are a computational biologist yourself, it's fine. If not, then connect with, make a good team with some computational biologists. So that will be one thing. Then identifying the problem, and if you can use some in-house resources like the Indian genome that DBD has recently released. If you want to work on that, that will be even better than using the data from TCGA or Caucasian population or other population. If you can get your hands on some in-house data, that will be even better. If you have got data from the university, from your own place, that will be more robust. There are specific calls by DBD. Now they have a section on computational biologies. Earlier they did not have that. So you can apply under those sections. They invite grants twice a year. So you can apply under that section, which is totally data driven, and it should be easier than putting it for the wet lab studies for genetic data. I hope that answers your question. And you can connect later on as well for more details about particular funding agency that needs to be tapped in for getting the fund. Yusra is asking what is the exam passing cutoff. If we just give me a minute, I'll tell you about the grade. So the exam passing cutoff is 35, and I've already shared this document which will give you all the details. So if you can see in the chat box. Yes, sir. I think, Swati, we are getting no more questions. So we can close the session here, and we reconvene at 2:45. A reminder email will be helpful. If we send a reminder email to all of them, the 2:45, everybody should log in so that we can start the exam at 3 p.m.
today, and I've been asked to present some recommendations, some thoughts on how we can streamline drug discovery through target identification in Indian context. So I think I'll give you a brief recap what we've discussed in the past 5 days. I hope now, having done this intensive, shall I say, boot camp training, you're more confident in your understanding of cancer biology than it was 5 days ago. Uh, I think what we have been able to do is show you some defects in tumors which actually can be exploited for the therapy of cancer. And I hope I've given you some flavors along with my colleague, uh, co-host running this, or shall I say the host running this, Dr. Aziz. On omics technologies, we touched upon some functional genomics. We discussed some aspects of CRISPR screens, especially I showed you the platform depth map. Also, we discussed some aspects of preclinical testing, how drugs are tested, and some challenges which I'll discuss if I haven't already. So I think we will all agree there are lots of strength in the Indian academic system with regards to cancer research. The soil is already there. There is, of course, very strong expertise in molecular biology, in immunology area in India, and what you would not perhaps realize how big is this initiative that government actually funds research fellowships. I'm talking about early career research fellowships given to students like junior research fellowship, senior research fellowship, and postdoctoral fellowships. They are uh, highly valuable in terms of providing the training. To give you a context, in British system, there isn't anything like that. Normally, the government provides funding as DTPs or doctoral training programs to the academics, to the faculties. Then they advertise it. Students are selected. So there aren't any students who come here, walk in the lab, and say, "Look, I have my PhD studentship generated through passing a competitive exam." So the system is already very robust. I think this is one of the unique strengths of the Indian system. Certainly, the provision of providing through, of course, competitive exams, which are highly competitive exams, actually, as you all know, providing these research fellowships. And I think there are not very many, but there are sequencing and bioinformatics capabilities in India. They could be, of course, they're developing at the moment. There are increased number of translational initiatives. I can give you one example. Tata Cancer Memorial Cancer Center has some collaborations with us in University of Surrey in the UK, and a collaboration with. So this is a triangular collaboration between us, Surrey, and King's College London. Sometimes we have conferences where we host people coming from Mumbai. So these initiatives are actually quite uh, pivotal in cancer discovery research. Um, and one of the major strength is, you cannot deny that, is the large patient population, and we have diverse genetic backgrounds. This could be a major resource in discovery research, in validation of the clinical findings. And why this is critical, there are lots of cases, and sadly, with every nation on this planet, the cases of cancers are on the rise in India. Uh, if we have a system where you can actually longitudinally follow up patients, what happens with their cancer, or in fact, before cancer hits, start early and collect those samples I was mentioning to you, blood samples, urine samples, prospectively to see what happens to these individuals. you can go back and have a look at their samples. Of course, our capabilities to identify biomolecules, metabolites is always on the rise. So we are getting technologically advanced across the world, and that would be quite good because there is certainly a very rich longitudinal follow-up data that you can uh, have access to. Sadly, like many economies, it is an issue with the patient receiving the right care. The drug development costs are enormous. You see, many drugs are, of course, all drugs. These are mostly Western-made drugs, of course, are patented, and that increases substantially the cost to the end user. Many times in countries like India, see government-led health economics, health systems, which actually bear the cost of this, but many times patients are asked to purchase drugs, which are often quite expensive drugs. Not many people can afford it. So if you have local innovation, I think that would be quite good in terms of lessening the dependency on Western made drugs. Uh, of course, there are initiatives to enhance the participation of patients into clinical trials, and this is something which is getting better in many nations, including India. At the moment, I think what needs to happen is to have biobanks, which are perhaps more scattered. It's very difficult task. Don't think it's only India to have them on a single portal where anyone, any investigator can come and access the tissues. For example, it's a mammoth task. And overall, this problem lastly exists not only in India, but many nations, including UK. Perhaps it's not as much here, but basically there is a lack of providing training where students are trained in wet lab techniques, for example, Western blot skew, PCR, DNA, RNA isolation, for example, but also to be able to analyze any omics data that came from that wet lab exercise. And therefore, there is always a reliant on, even for basic things, basic analysis on clinical trials. Can you hear me?
Yes.
Sorry, Dr. Aziz, you were mentioning the other day you lost internet connection. That happens in the city of
Uh,
Cambridge in the
it
United Kingdom
happened,
as well.
it happened with me today as well.
Sorry, I can't hear you.
It happened with me today as well.
Hello.
Yes.
I had to connect to my phone. Okay, thank you. So yeah, Dr. Aziz or anyone else, please jump in to complement this informal lecture session. Uh, you can see my slides again, right?
Not yet. Not yet.
You cannot. Okay.
In my concluding lecture today, I also talked about a few summaries and
Uh-huh.
recommendations. And one of them was about Aligarh Muslim University contributing more towards cancer research. Plus, yesterday
Sure.
there
Yeah.
were results for the Anusandhan National Research Fund grant came out, and there was a
Hmm.
blood. Very,
Really.
very few people got the grant, and even the review process was not satisfactory. So now we will be getting the full reports. I think this is a
Okay.
worldwide problem about funding, so that's why we need to find alternate ways to find
I, I
how to
think
get.
that is a scenario worldwide. There is less funding for biomedical research through public means. Mm. Can you see my slides now?
Yes.
So I think why we need indigenous target discovery initiatives in India is also because, of course, given the genetic diversity or genome, actually is not really, you cannot really rely 100% on drugs being developed for Western population to work as efficiently in Indian context. And for an example would be mutations in genes, tumor suppressor genes, just such as P53, TP53 differ based on what geographical region you hail from. And that's why it is very important to lead these efforts in India through our indigenous systems. They will, of course, be very affordable compared to drugs being developed by big pharma in the West, and actually can be quite good for Indian health systems as well. Yeah, perhaps we don't realize, but the cost of health system, especially if it is public funded health system, the cost to the economies, to the governments are enormous to bear. Uh, so what I think could be the major challenges, I may be wrong, these are only some ideas, so please feel free to jump in and correct me. Uh, I think one of the things that can happen is the centralization of resources, especially development of data repositories, not to work in silos. That would certainly help the drug discovery efforts if we have platforms like omics platforms in one place. I think, and this number two is not a problem only associated with Indian context, perhaps, but even in the Western countries, it's very difficult to find biologists, especially working in cancer research, to have active, fruitful collaborations with clinicians and computational biologists alike. And that is a big challenge, certainly, in terms of integration of the interdisciplinary approaches. So that's difficult. From what I realized, and as I said again, yes, you have a question, Dr. Aziz?
Yeah, I just wanted to add here that I talked in my previous lecture that this limited interdisciplinary integration can be supplemented, and we have already done it successfully by the consortiums, where you
In fact,
bring
yeah.
together the expertise on one platform. So this can be done. And I learned it from being in the US, where different institutions all across US were coming together to work and address this problem of cancer. So at least we have a solution for this number two, limited interdisciplinary integration. If anybody is interested, we can teach them, we can tell them, we can tutor them, we can mentor them.
I think these consortium led initiatives that you're coordinating, heading, they're certainly very fruitful for this, and I think students should really think about joining them. Uh, from what I understood, the point number three is there may not be very vast exposure to already publicly available data sets to especially students, despite them being publicly available to every investigator across the world. So perhaps that can be something that perhaps we are not very well trained with in India to make use of them. Certainly, that can help if we have any, you know, satellite meetings or such initiatives. Um, that would certainly help. With regards to grant culture, I don't. Are there any systems like RO1 system where you can get renewable funding every 5 years based on your performance? Uh, that would certainly help if you know that you have a sustainable funding. It takes time, even if once you have all the data um, at your desk, it takes time to publish those papers. Preprint is there. Not many people feel comfortable placing their work on preprint servers, but it's still it takes a long time, and unless and until you're sure that you have a secured grant funding, uh, not many people will want to invest in something which is not going to be, let's say, giving you any outputs in the next 5 years. So there has to be some sort of a stream where that there is a sustainable grant funding system to support my research. Uh, in terms of this number 5 point, I think with regards to animal models, we need to, most of the models, cancer models, cell line models that we use are mostly Caucasian based, especially in US, where, for example, prostate cancer is very common in Afro Americans. We don't have very many popular, freely or shall I say, readily available cell line models from Afro-American populations to be used for studies in cancer research. Uh, Also, as I mentioned, there are skill gaps. If you're a computer, if you're a biologist, you're a biologist. If you're a bioinformatician, many times in Indian context, perhaps the exposure, even in countries like UK, when I speak to my colleagues who are computational biologists, they do get excited with the developments that are being made in biological research. And we ask them questions: how can we look at this on a high level from a bird's eye view? And that's where their role comes in. So sometimes I realize they are not really they know biology, they're interested in biology, but unless they have a biological input, unless we have that complementation, they are incomplete without biologists, and vice versa. So that certainly will help if we can shrink this gap. Uh, what I would also suggest is, of course, to strengthen infrastructure. If we can invest uh, in high throughput screening facilities where at a fraction of cost funded by the government, you can get your samples analyzed based on a strong rationale. So the government centrally funds this. Um, Also, what would be important is to do capacity building. That is what Dr. Aziz, through his consortium, is doing. This is one of the excellent examples I have. Of course, don't work in isolation. So if we can break silos, science is not done in isolation unless you are in the industry. You don't really want to disclose your research and compromise, jeopardize the invention. But certainly, as academics, we should certainly have an outreach ability to go and talk to people who we think are like-minded and who will want to complement the research through collaboration. So that certainly helps. And of course, one of the things you can do is to reach out to partners in internationally. That certainly helps. There are people who are very busy. They will tell you, "Sorry, I don't have any bandwidth to collaborate." Others will be more than happy to collaborate. So certainly, being able to reach out to people, and in this world of where you can simply write emails to people, that would certainly help. Um, of course, point number three, development mandate open science practices. This is mandatory in terms of many journal policies. You're publishing something, you will have to provide codes. You need to provide fully the protocols which can allow other investigators to be able to reproduce the results that you have published, for example. And what I would suggest, if you have any DTPs or doctoral training programs, this is something that we can start in India, uh, and especially to focus on cancer research. And in those cases, I think one of the criteria should be to have at least identified some targets in order to receive a successful PhD degree. So that is something that can be integrated in the curricula itself, and that would be certainly very helpful. And in terms of funding, I think the problem we have is that, and this is not the Indian problem, there is no one who would want to fund high risk research. And even if they say, sometimes for high risk research, you don't need preliminary data. But sometimes when you're reviewing such applications and you realize at a later date they have been successful, turns out they have tons of pages of preliminary data. So there is a bias in human nature, I think, where we want to mitigate the risk and make sure we don't really support high risk projects, which often should not have required any preliminary data. But that is certainly not the case. Uh, what I could also suggest is to development, like Dr. Aziz is doing with his consortium, development of a cancer omics portal where you can have Indian cancer genome data, proteome data, for example. There have been some initiatives, I'm sure, in India already to have number two, this screening consortium where several institutes like AMU, uh, where we have few good investigators working on cancer research can actually come together, collaborate with industries and other partners, for example, providers of high throughput screening, and form international consortia. Perhaps this is already happening, I'm not sure. And of course, to develop open source tools. For example, what Dr. Aziz mentioned yesterday about not being able to access IPA for data analysis, omics data analysis. This is quite alarming. To tell you the truth, it won't take more than 4 computational biologists and 1 month of their time to develop such tools, which could be publicly available, similar to uh, these platforms, which you can only access by having a paid subscription. I would certainly think about that because most of the time we are using for data visualization, we use R in the lab. We have local colleagues who support these, which we collaborate with. So I would certainly think if we can make something which is available to everyone. We already have some tools at the moment. They're not published, therefore I didn't show them, but we already have developed some bioinformatics tools which will be available to everyone. And I think one of the problems I don't see very many industries in India who are very keen to develop, take forward new target discoveries, and uh, work on developing new molecules to target a new cancer target. Mostly they're making generic drugs, and so on. So I think what would be really good is if there are some genuine interest from the pharmaceuticals, Indian farmers, to take initiatives to develop new drugs against the new targets, especially which are more relevant in Indian context. And then for the academics to reach out with them and establish collaborations, certainly through funding from industry. Uh, I think what is important for any research, this is not in particular applicable solely to cancer research or target validation, is to create a more fellowships program. We have a problem in countries like India, where due to limited number of opportunities, there is a lot of brain drain. And if we have attractive fellowship programs that guarantee an academic post, for example, or something equivalent, which can allow to attract academics back in Indian academic ecosystem, then certainly this will help with the acceleration of research. Courses like Gyan are excellent, and thanks to Dr. Aziz for organizing two courses back to back, one on drug discovery and this one here that we are concluding today. These short courses, workshops are certainly going to be very, very helpful in the development of local population capacity building. Uh, and I think with regards to the career tracks for academics, teaching facing academics, research facing academics, for example, myself, I do teach and I do research. Something like that would certainly help, where you can actually pass on the benefits of your research to not only people who are doing PhD postdocs in your lab, but also to the students who are yet to decide. They're doing their masters, their bachelors, they're yet to decide what to do. And if they have their faculties and their teachers teaching them discoveries which are homemade, done in local universities, I think that will certainly feed their enthusiasm. And of course, what we do here during the BSc, many students will do industrial placements. So something like that, where PhD students, at least to start with, uh, can go to an industry for a year, take a year out, and learn how cancer research, screening especially, is done in industry. So what I think, and forgive me please if there is something which is out of line here, but I think for faculties, if we have interdisciplinary teams for grant applications, you certainly enhance the chances of getting funded. Science, as I said, is not done in isolation anymore. It is become certainly highly, highly interdisciplinary. So if you have teams, if the grant funders can see the evidence that you're proposing something which you can deliver through these collaborations, and they have the right experience, then certainly there are more chances to be getting funded than proposing something where you're totally out of your bandwidth. You don't know what you're talking about. How would you achieve this? That would be super difficult. For students, I think what would really help, especially those students who have been very good in mathematics, is to learn coding, programming languages. For example, Python for automation, pipeline studies, development of pipelines for high throughput data analysis, and especially R, which is very important for statistical analysis. I think we should certainly have core facilities. I understand it's very difficult for each university, but at least like-minded universities within a geographical region may have some shared core facilities. Uh, and I think where you can send your samples and do it at a fraction of a cost, including the initial analysis for sequencing, for example, imaging, mass spectrometry for proteomics, and computing capabilities. And then for institutions, it is important that we reward open science and certainly reproducibility, which is a big issue, as you know, in science, in promotion. So this certainly helps with promotions. Academics will be more triggered to do research. They can see the benefits of it. And for policymakers, I think having niche initiatives to promote target identification research to start with such programs would be good. I'm not sure how realistic it is, what I've written, but also we can establish fellowship programs for younger research in target discovery. Um, I think to start with short funding, as I mentioned, 500 crore to start with, start some training boot camps that can also then support development of biobanking, where you can access Indian cancer tissue through tissue microarray biobanks. Then to certainly support the validation of targets using those samples rather than relying on samples from Western countries. You can buy these days tumor microarray chips slides, but mostly from Western nations. Uh, and then certainly that will help, of course, the policymakers to see that will help policymakers to see the develop confidence that actually this research actually does provide good return on the investment, and they will be much more keen to support such initiatives, and perhaps can also help at development of at IND stage, where the patents are filed post preclinical data, and that will certainly help with establishing stronger industry pipelines. These are some of the metrics of success. I think if we have what I did mention, there will be certainly uh, the governments, of course, would want to see what have you validated, what are the reproducible targets, are there targets which actually can be applied to pan cancer initiatives, or there are targets which can be validated across different population, across different human races. And certainly what will help is to get more number of patents based on these discoveries as an output, as a showcase of output from the work, from the investment that the governments do. Publications certainly help, and of course, the number of trainees which have been certified to use a specific platform, so that will certainly help. With regards to the clinical translation, I think biomarker-led trials can be initiated within Indian context, and as I said, what would certainly help is based on the discoveries and intense target identification initiatives. If there are more submissions, and then if we can get licenses um, for drugs, worldwide licenses sometimes, that would certainly give a much better, much substantial return than the money invested. And certainly that will attract not just a patent, but certainly something which has been licensed to a pharmaceutical industry, be it national or international, would certainly interest the policymakers to invest more money, of course. And of course, lastly, this equitable access, where we know that there are geographical inhibitions, there are areas which are totally desert, there aren't any top institutions. We should certainly establish institutions in those areas to promote academic research, um, and not just favorably fund certain institutions. And with this, I would stop and take any questions if there are any. Yeah, I'll have a look. Let me check the. Okay, so let me go and take a look from the beginning. I'm seeing. Uh, I think what I would say, if I understood the question correctly, forgive me, I can't read it, so looks like a long question. But what I would say is certainly there are chances to activate, uh, reports, at least proof of concepts, where you can reactivate tumor suppressors. But that will again depend. How would you want to do it? If there is a genetic loss of tumor suppressors, there is no way to reactivate. If there are any specific point mutations, then certainly molecular docking will help to screen, the in silico computational screening to see basically what sort of molecules will bind and engage probably trigger activation of this tumor suppressor. Certainly, that will help. That's a good thought, Aparna. I don't see any more questions. No problem. Swati, sorry. Are there any more questions? Ilma is saying, please do not mention our grades in the certificate. Yeah, it's totally up to Dr. Aziz. I'm not. I've only provided part of the questions. That's up to him. But I'm sure he's not going to share everyone's marks with everyone, so don't worry. Swati Afsha is saying she hasn't received any uh, link for any test. Okay, all right. Okay, how come I'm not able to see these? Let me check. What time did it come? Okay, that's good. Okay, okay, okay. Yeah, I can see that now. Thank you. Uh, what are the limitations in targeting NTD as a drug, as a drug target? I guess Dr. Qureshi is talking about the androgen receptor. I would say one of the major limitations is that it is one of the proteins which remains amorphous, meaning it does not really fold into a solid, tight, three-dimensional structure, given its intrinsically disordered nature, which is that directed by the amino acids which it is formed of. This is not just with this protein, but many other proteins. Unless it can be something which can be crystallized, it is very difficult to then dock molecules and see what will bind to it. Dr. Qureshi, if that answers your question. There is a question from Dr. Juhi Gupta. Examination link will be shared in chat box. I'm not really sure. Thank you. Likewise, it was a pleasure to be here and contribute. I can see the. Thank you, Swati. I can see there are lots of people now for. So the test is starting in 19 minutes,
Yes, yeah.
and it is for an hour, right, Dr. Aziz?
Yes, it is for an hour. And I would like to personally thank you. And if you want to hang around after the exam, we will be having just some concluding remarks. But it's totally up to you. And we'll be in touch again soon.
Thank you. I'll try definitely to be here uh, for that. So the exam will start in 20 minutes now. If any one of you has any remaining questions, want to switch on your speaker and ask me those questions, please feel free to do that. Thank you, everyone, as well. Swati, is it possible to get some? Is there any that the students and any participant can provide an anonymous feedback on the sessions that we held here today and for the entire week? Happy to have them as anonymous because I think unless they are anonymous, there is little point in getting any feedback from any participant. So, is it possible, Swati, to get anonymous feedback or not really? Okay, that's fine. No problem.
Well, in Google Forms, it's not possible because you have to log in with some things. But
I
yes, we
understand. Okay,
cannot, we'll remove that thing that collecting the email address. We'll not collect the email address. Then it
okay.
can be anonymous on
I
our
think
end.
it would be great because that certainly helps us, Dr. Aziz, myself, to
Mhm.
actually going forward improve and
Yes.
remove any deficiency. So I think we as academics rely a lot on the feedback that we receive, good or bad, equally helpful.
I totally agree and I understand. And I will appreciate if everybody does do their Google Forms for the feedback and they don't have to put their email address.
That's great.
Okay. See some email addresses being typed here. It's for the exam purpose. Azam, we already have these email addresses. Can you just add them and give them access? So we'll shut down the meeting and then we'll allow the only the yes with their registered email IDs that you already have given access to, right? Okay, thank you. Okay, okay, if they have not received an email, that's a different story. But they should check those emails that were registered for your for this course. I think everybody has been sent this. Usually, what happens, they log in from some other email and then they have given some other email for the registration. To use the same ID that you have used for registration for checking your emails and for logging in. I think the exam is coming to a close, and Swati, you may please take over once you close the exam. Thank you, Swati. And I think we don't have enough words to thank everyone. But before I conclude, I would love to hear from Dr. Asim if he has some final words of wisdom for all of us. He being our foreign faculty member, the entire course was possible because of him, because the GAN course starts and stops at the foreign faculty name. So, Dr. Asim, would you like to say a few words before I say the concluding remarks?
Thank you very much, Dr. Aziz. It's been very kind of you to host me. I wish it was possible to come in person, but yeah, things beyond our control, I couldn't do that. It was a very good experience. I am deeply impressed with the quality of participants who were attending the course, and of course to all the members, your staff, who actually did take care of everything painstakingly to make sure that the course ran on time and streamlined. So thank you, everyone, and I wish you all the very best, all the participants and everyone else in your future career. Thank you very much.
So thank you once again, everybody. And we say that we keep the best things for the last. So I would love to acknowledge and appreciate and mention the efforts that were going on in the background. I would love to tell the support that I got from my family, especially my wife, Kanata, my kids, and my family members who stood by me, who stood by my varied schedules, the late night and the early morning schedules. Thank you so much. I can't say enough to express my feelings here, but you have been a constant support to me. And my family members and the entire family of the Interdisciplinary Nanotechnology Center and this university, which I'm trying to passionately serve as much as I can. So wish you all the best, all the participants. You will be getting your certificates soon, and see you again sometime. Bye-bye.
Bye, everyone.